So with that, I'd like to move on to the next session. In the interest of time, I'll very briefly mention who's going to be in our next panel. Again, I want to emphasize that the bios are available on the website. So go ahead if you want to hear more about each of our esteemed colleagues and speakers, go to the website and look up their bios. So quickly, we're moving from the genetic scale to a little broader scale, which is the physiology and microbiome scale. And so we're going to kick off with Sarah Berry, who's from King's College London. Then we're going to move on to Michael Ryan from the Weizmann Institute of Science, and then finish up with Guru Banavar from Viome. And then so each of them are going to give a 5-minute presentation introducing their technology that they've been working with. And then we're going to bring everyone together to have a discussion. So again, if any audience members, if you have any questions, feel free to send them along. So we'll try to get to your questions as well. So without further ado, pass the virtual mic to Sarah.
Great, thank you very much. Hopefully you can see my screen. Oh. Bruce, thumbs up, you can see everything?
Yes, we do see your screen.
Great. Okay, fabulous. So thanks for the introduction, Bruce. As you said, I'm a nutritional scientist at King's College London. Quickly, my conflicts of interest is that I have received research grants from the Almond Board of California, the Malaysian Health Ministry, Zoe Global, and the UK Research BBSRC Research Councils. And I also receive consultancy from Zoe Global. So I'm going to talk today in 5 minutes, if I can, about the PREDICT programme of work to illustrate how we've utilised big data and novel technologies and really kind of pushing the fast forward button in nutritional research, and also the creation of a personalised nutrition programme, which I think feeds back nicely to Mariatte's talk earlier today. And what we've been doing with the PREDICT studies is really moving beyond the constraints of traditional nutritional research, where we've had to compromise between either quantity or precision, for example, with either high precision but low quantity RCTs or low precision but high scale epidemiological studies. And we're really capitalizing on these big data opportunities that we have from novel technologies such as digital devices, clinical devices, remote clinical testing, but really importantly also from citizen science, which is providing us with data of scale, breadth, depth and precision, which is really becoming the focus for us within our PREDICT programme of research. And we've done this within a really unique research collaboration, which is between traditional nutritional academia at various institutions, including King's, where I'm based, but also Tufts, Harvard, Stanford, and MGH, and also a startup tech company called Zoe, who specialise in machine learning and AI. And this enables us to conduct this really large ongoing programme of personalised nutrition research at this scale, breadth and depth that we just wouldn't be able to do in traditional academic setting. Now, the programme consists of a number of studies. There isn't time, unfortunately, to give an overview of all of these studies. I'm going to briefly cover PREDICT 1 as a way of giving you an example of the kind of methodologies we're using and the kind of results we're generating. If you are interested, you can get full details on all of these studies from the Zoe website, which I put down here in the bottom corner. What is common, though, to all of these studies is the use of novel technology and remote home testing. Also, what's common to all of these is the measurement of the many integrated and the interrelated pathways within the same study that determine how we respond to food. So we're not just focusing on single exposures, single outcomes, as we often do in nutritional research. An example of the type of methodology we've been using is shown here from our PREDICT 1 study, which has provided the foundation for our machine learning algorithms that we're using within the Zoe personalised nutrition product. It consisted of 1,000 participants in the UK, which was our discovery cohort, and then we had a validation cohort of 100 participants in the US. This study involved a clinic and a home testing phase. During the very tightly controlled clinic day, we provided sequential test meals. We collected blood at regular postprandial time points for detailed metabolomic analysis. And alongside this, we also undertook genomic testing. We collected stool samples for metagenomic analysis, as well as measurements of anthropometry, diet, and health. And then participants entered the remote phase. And this is very similar, like I said, to the Zoe personalised nutrition product arising from this research. And this phase consisted of a 2-week at-home period with standardised as well as free living meals, during which time we measured glucose using continuous glucose monitors. So using these remote digital technologies, we collected dry blood spots for C-peptide, a surrogate for insulin, triglycerides. We measured activity and sleep using activity monitored. Most importantly, everything that the participants consumed and their satiety levels were logged using a new dietary assessment app that we developed. And this was monitored in real time on a dashboard by a team of nutritionists. Again, harking back to what Mariette talked about earlier about combining these novel technologies alongside actually that personalised support. And this allowed us to collect really high quality dietary intake data. And this is an example of the kind of scale of data we've got just from Predict1. We now have nearly 10 times the scale of data with our Predict2 and our Predict3 studies, which really allows us to unravel the different exposures related to the huge variability that we see between individuals. And this is really nicely illustrated here from our Predict1 study. The full results of this were published last July in Nature Medicine, if you are interested in looking in more detail. But this shows, I think, really nicely the huge variability that we see in individual responses in both triglyceride and glucose on the left and the right, respectively, in a 6-hour period after consuming a standardized meal in a very tightly controlled setting in healthy subjects. But you still sort of see this huge variability. And the red and the blue lines are the mean and the medians, just so you can see how in this context, it's really not very meaningful, this mean response to the majority of the people. And with all of this data we've been collected, we've been unraveling the impact of the many different exposures, and I've listed just some of these that we've collected here on the left and their relationship with the many different outcomes, and I've listed some here on the right, many of which are multi-directional. And kind of to give you a very broad overview of the results is that we found that what we eat, how we eat, so time of day, sleep, etc., as well as who we are, so our genetics, our age, etc., are equally important in determining how we respond to food. For each outcome, what we've done is we've also explored the relative contribution of these different exposures. And these are shown here for glucose on the left and triglycerides on the right. But what's noteworthy is that the importance of the different exposures, whether it be genetics or meal composition or age, for example, are different depending on the outcome. And this applies not just to glucose and triglycerides, but for all of the outcomes we've looked at, whether it's satiety, whether it's inflammation, for example. But ultimately, we want to use this information to advance nutritional science as well as provide dietary advice. So Zoe have used their machine learning expertise alongside this science to create an at-home test that allows us to predict individual responses to food and deliver this very personalised dietary advice. And the microbiome also forms a large part of our Predict programme. Again, there isn't time to go into all of these results. We do have a paper that came out again earlier this year in Nature Medicine. But in summary, we identified a diet health microbiome signature of what we call bad and good bugs loosely, which are associated differentially with bad or good clusters of both diet and health. And this allows us, alongside individuals' personalised microbiome analysis, to provide personalised gut booster or suppressor advice. So what's the future for the Zoe Predict programme? We're sitting on a goldmine of data. We're only just really scratching the surface of all the fantastic breadth and depth of data that we have. And we're at a point of moving from this very general up to this more stratified and personalised nutrition phase, and particularly with the introduction with the Zoe programme, which is the commercial product arising from this research that's really taking all of these findings and delivering this personalised dietary advice based on these many interrelated and integrated pathways. Thank you, and I hope that covered enough in the few minutes that we had.
So thank you. I'm very excited to be here. And so my talk today will focus on a unique approach that we developed for personalized nutrition by prediction of glycemic responses using clinical and microbiome features in different populations. And here is a short disclosure. And so I'll start with a study that we conducted 9 years ago, led by 2 former PhD students. And in this study, we deeply phenotyped 1,000 healthy individuals, including continuously tracked the glucose levels and collected multiple measurements such as clinical, lifestyle, diet, microbiome, and others. And we basically asked ourselves, what is a healthy diet? So to answer this question, we used a quantitative and easily measurable marker of postprandial glucose response, which is the change in blood glucose levels in the 2 hours after eating a meal. And importantly, it was linked to metabolic diseases, diabetes, weight gain, and lipid profile. And so our data of thousands of postprandial glucose responses to meals from that study revealed that different people have different responses to the same meal. And while within participants, as you can see here, the responses were reproducible, between participants in the different colored line were in a much wider range of PPGRs. So then we devised a machine learning algorithm that accurately predicted personalized PPGRs to any food combination. And we used clinical and microbiome data for this. And we further showed in a short-term intervention that personalized dietary intervention based on the algorithm that you can see here in the green line successfully lowered glucose levels in people with prediabetes. So next, we wanted to test what is the clinical impact of personalized postprandial targeting diet in population with imbalanced glucose tolerance, since this population can be benefit from this dietary approach. So for that end, we conducted a randomized clinical trial in 225 prediabetes participants, which were randomly assigned to one out of the 2 diets. It was either PPT diet, which is postprandial predicted targeting diet, or Mediterranean diet, which is the commonly recommended diet for prediabetes. And participants were instructed to follow a non-caloric restricted diet for 6 months, and with additional follow-up for another 6 months. And they were also connected to a continuous glucose monitor and logged their food intake during the whole study in a dedicated app. And at the end of the 6 months intervention, we found that PPT diet led to a decrease in daily time with high glucose levels and also a reduction in hemoglobin A1C levels as compared to the Mediterranean diet. And we concluded that PPT diet improves glycemic control and metabolic parameters in individuals with prediabetes. And following of this intervention, we did a pilot study in 16 diabetes participants without any glucose-lowering medications of 6 months of a PPT diet, and we found that changes in gut microbiome were associated with clinical outcome. And for example, changes in Eubacterium ventreosum were negatively associated with changes in fasting plasma glucose levels. And we saw in another study that suggested that lower levels of this species at baseline were predictive of improvement of insulin sensitivity after FMT in patients with metabolic syndrome. And additionally, we saw that PPT diet led to a reduction in hemoglobin A1C levels. And in general, PPT diet induced diabetes remission in more than 60% of these participants. And lastly, I want to present an ongoing multi-center dietary modification project in individuals with diabetes and glucose-lowering medications that done by Day 2 company and who has a license to use the algorithm that I just presented. So in their intervention, they showed that following the diet led to decrease in hemoglobin A1C in more than 80% of the participants, and also a reduction in body weight, and also 30% of the patients reported of a reduction in the use of glucose-lowering medication after 3 or 4 months of intervention. And in summary, I just want to say that these studies support the idea that personalized dietary interventions can potentially improve glycemic control and metabolic health. And I want to thank my team and collaborators, and thank you for listening.
Hi, everybody. My name is Guru Banavar. I am the Chief Technology Officer at Bio. I'm also leading a whole program in Discovery AI. My background is in AI and applications to many fields, including systems biology. In terms of disclosures, since I work for Viome, I'm going to be saying a lot about what we do at Viome. So that's that. So to begin with, my two previous speakers have provided a very good summary of how DNA can be the basis for understanding glycemic response and some other metabolic impacts on biology. What I want to say here is that it's not just the genetics, but the gene expression, that is the mRNA molecules that are produced, transcribed by your genome, that controls chronic disease at the end of the day. And that, the gene expression, can be modulated with diet and lifestyle. So if you want to take an example, take C. diff. C. diff colitis is a very common disease in the US and in the rest of the world. But if you look at some of the underlying symptoms and underlying phenomena, Clostridioides, which formerly was called Clostridium difficile, the presence of that organism does not mean that there's an infection. The infection begins when that organism starts to express certain genes that are going to end up being toxins for the human body. Similarly, on the host side, the same healthy colon can express very different genes when there is such an infection. So you see on the left side, a healthy colon, on the right side, a very sick colon. And the difference between them is gene expression, not the original genetics or the DNA. So the point here is both on the microbiome side as well as on the host side, what is important is the gene expression rather than the original genetics. So with that observation and premise, we at Viome have created two key technologies. One is metatranscriptomics. So you've heard of metagenomics, but this is metatranscriptomics, which is the measurement of and detection of mRNA molecules across a whole range of organisms, from microbes all the way to humans. And that is the process by which we generate data in Viome. We have a scalable platform for generating mRNA data that can be then used by our AI and machine learning technologies for deciphering the insights and the processes underlying those molecules, as well as continuously learning from real world data. So those are the two key technologies that we are employing in Viome. So using those two key technologies, we are recommending food and supplements. And again, remember that this is mRNA, this is the gene expression or the activity rather than the genetics itself. So if you think about foods, and you've already heard about how we can make food recommendations using the knowledge of the biology, the genetics, what we have shown in our study, which is essentially very equivalent, and in fact, I would claim quite a bit better because we are using genetic expression, mRNA molecules, we essentially show that we can predict glycemic response at the state-of-the-art levels. And we can, in fact, show the differences between two foods for two individuals being opposite, depending on a lot of biological factors, including the gene expression in their body. And we can classify every single food as enjoy or minimize foods. And furthermore, we take it one step beyond the glycemic response. We look at a lot of pathways in the microbiome that are essential for our health. For example, short-chain fatty acids. Butyrate is a great example of that. We look at the production of butyrate in the gut microbiome. There are organisms that are, more importantly, not just organisms, but the functions, meaning the genes and clusters of genes that are responsible for generating butyrate, and other kinds of short-chain fatty acids like propionate and so on and so forth. But the gut microbiome is not only responsible for generating pro-inflammatory beneficial metabolites like butyrate, but it's also responsible for eliminating things that are not very beneficial, such as oxalate. You need the microbiome to eliminate oxalates. And you also need, you can look at other processes like sulfide production and so forth. So taking into account all of these pathways that go beyond glycemic response, we then take the output of our glycemic response predictor and classify all our foods for a given individual into 4 categories: superfoods, enjoy foods, minimize foods, and avoid foods. And there's quite a bit of research and science that has gone behind this level of recommendations for foods. As an example, if you take spinach, which everybody knows is a very healthy food, right? And so it probably has very low glycemic response. So it would normally be an enjoy food for everybody. So we further look at the oxalate elimination. Spinach is a very high oxalate type of food. So we look at that micronutrient and we look at the oxalate elimination pathways. And if the pathway activity for oxalate elimination is high, then spinach can be a superfood for an individual. But if it turns out that if the oxalate elimination pathway activity is low for an individual, it can be deleterious to your health. It can in fact create kidney stones and other kinds of complications. So if everything else is equal, we would want a person with low oxalate elimination pathway activity to avoid spinach and avoid those kinds of deleterious health effects. And so we have hundreds of pathways such as these that we evaluate and we have studied, and we are publishing those results one by one. So we take this whole system biology approach, if you will, and look at what are the underlying pathways that needs to be taken into account for recommending specific foods to specific individuals based on the gut microbiome activity. Now, we have a consumer-based product which currently has approximately 250,000 samples of lots of customers around the world. And we use all of this data to come into our AI engine, through which we not only get the food recommendation insights, but we also are able to detect certain key initial signals of chronic disease. And we've started an entire discovery program around this. And we started to look at many therapeutic areas: metabolic diseases, type 2 diabetes. We have a publication that's just coming out for determining both a risk for type 2 diabetes, like prediabetes and insulin resistance, but also looking at whether microbiome plays a role in the metabolism of drugs like metformin, as an example. We look at gastrointestinal diseases, neurological diseases, and so on and so forth. We also look at rare diseases, rarer diseases like oncology, and we just recently got an FDA breakthrough device designation for a biomarker that is useful for diagnosing very early stage oral and throat cancer. And so that's an example of the kinds of the power of mRNA-based discovery using very large-scale data. So here we are talking about hundreds of thousands of individuals which have a range of different chronic diseases. And we have many, many clinical studies that are all looking at these entire portfolio of diseases. So if I look at the future, that takes us to a world in which we can study host microbiome interactions, not just the microbiome activity, the organisms and the functions of the microbiome, but also how is the host responding to it through the immune system, through the inflammatory pathways, and so forth. And we have this platform through which we can detect the molecular functions for the microbes as well as the humans and the interactions. And so we have just renamed ourselves to Viome Life Sciences for taking the systems biology approach. So thank you very much. And I think we can open up for questions.
Excellent. Well, thank you, all three of you, for some very thought-provoking presentations. Again, we understand that you could very well easily speak more than 5 minutes on each of these things, but we wanted to really seed the discussion. So wanted to start off with the question of bias. One of the concerns that's being raised when it comes to different, certainly any type of research methodologies, but also with computational approaches, AI approaches, machine learning approaches. What types of biases do you see potentially creeping into like the methodologies that you described, and how do you mitigate those? How do we really approach those?
Bruce, I can get started, and I'm sure the others can add as well. I think one of the key approaches for dealing with bias is to essentially increase the sample size and to ensure that the design of the study incorporates a very large collection of different phenotypes. So one of the key advantages is, when you look at most medical research, it's a few hundred, maybe a couple of thousand samples and so on. But when you get to 100,000 samples, right, and you are capturing phenotypes from 70 or 80 countries around the world, I think you end up getting a lot more representative data set for across the entire human populations, and that fundamentally, I think, adds to reducing bias. There are many other techniques as well, but that's one I wanted to highlight.
Mm-hmm. Sarah and Michal, any initial thoughts about that?
I mean, I would just agree with everything that Gary said, that I think, the problem we have, and this is a problem across all nutritional research, is that moving away from the worried well that tend to either volunteer for nutritional research studies or are purchasing personalized nutrition products. And it's a case of, as Gary said, is scale and multinational studies as well.
And also agree, and we now conducting other studies all over the world, international studies, and also taking a lot of data and continuous data to try and support the real data without a bias.
So one of the issues is when people see, so all three of you described in good detail in terms of the data sources that you used, but there are a lot of other situations out there where you might hear, oh, okay, we applied machine learning to a data set and these are the findings, et cetera. Should there be particular stipulations? What should people be looking for when it comes to, understanding what biases may be going into the results that are being generated?
So my view, Bruce, is that real world evidence collection is super important. In other words, when you do the initial training of a machine learning or an AI system, of course, you are limited by the training set, right? But I think there has to be a way of continuously learning and updating your model and your recommendations over time as you increase your data set, right? So this continuous learning approach through a real world evidence that is robust and that can be shown to be better than the previous versions, that is a very essential cycle. And I think even the FDA is now basically providing frameworks for doing this kind of real world evidence data collection.
Mm-hmm.
I think as well, given the nature of as Gary said, the way that we're continually building on the data, evolving, it's also as researchers, scientists or companies being open to changing advice, having the courage almost to say, actually, the advice we had before was based on evidence that now we built on and we've got even better advice. And it's having that courage to evolve your advice as well and evolve your algorithms, your learnings.
Mm-hmm.
And also more deeply monitoring devices that help us to have more correct data.
And Sarah, you raised a good point that there is a certain segment of people who may be more likely to be participating in studies or offering their data and information. And one of the important themes throughout this workshop is, how do we set up a situation where there is appropriate diversity and appropriate equity, and we don't really maintain or even potentially worsen disparities in health. Do you have thoughts in terms of how to, the people are going to volunteer, the types of people who are going to volunteer or the types of people who are going to volunteer? So how are you going to, how would one actually change the type of data or the sources of data?
So I think there's a couple of points there. There's one, what's the kind of big end goal that hopefully we want to be able to make personalised nutrition and personalised approaches available to everyone. And then there's the other question of how do we in the short term make sure that we're collecting appropriate data that then we can build personalised advice across a whole broad spectrum of the population. I think for the first one, it's about, thinking of novel approaches to recruiting people, as we do with our nutritional research, making it as accessible as possible. But we have to be realistic as well, is that, this isn't going to be accessible for everyone. But by getting, as Gary said earlier, as much scale in our data, even though we might have only 5% of people or 1% that represents a particular ethnicity or age or etc., that actually at scale that still provides us with enough to start to get a hint of what's going on. And I think the other question about the broader application of personalised nutrition, which I from what I understand might have been discussed more yesterday, we're at this transition where, yes, it's exciting, yes, we're creating all of this high scale, high depth data, and we can apply it still in a stratified way, and that can be accessible to all. But by us learning more and more, then I think that we will eventually be able to understand what key factors are important based on a few key basic attributes of someone to be able to offer that more personalised advice in the long term and make it accessible to everyone.
Bruce, can I just bring up the economic issue here?
Sure.
RNA sequencing has been very expensive. A lot of the academic institutions have not been able to do it because it's been very expensive. One of the key breakthroughs that we've made in Viome is to bring the cost of RNA sequencing, which is much more advanced than, sort of normal shotgun DNA type sequencing, down to the level where most consumers can, or at least in the United States, can afford it. We're talking about $100 or a little more than $100 to get to a test, but I think the cost is going down dramatically. So in the next few years, I definitely think that we should be able to expand to developing countries and to get to the point where most of the, let's say the middle class population in the world should be able to afford and get the advantages, the health advantages of this kind of technology and this kind of personalization of precision health. I think in the long run, I can imagine that the cost of processing samples and providing this kind of biological insight should come down almost to zero, so that the real value for people is going to be in the benefits they get, like the food that they buy or the health benefits that they get. That's where the value is going to be. And, I think we need to shift it from a cost question to a value question to even the middle class population of the whole world. And that's sort of the strategy of Viome.
If I can also, thanks. So I can give an example from what we do here in Israel. So Day 2, the company, I presented their results. So they went into the health service of Israel, and they can give more low cost personalized nutrition, and they also use the dietitians in the health service. So it's also a combination of the technology with the dietitians here, which is also very important.
Excellent points. So we have about 2 more minutes before we have to move on to the next session. So I just wanted to address a couple questions that are very similar that came from the audience. Both of them asked, looking at the combination of foods. So they emphasized that, if you have, for instance, postprandial glucose excursions based on a single food, that can be misleading. And how do you consider things in the context of a complete meal? Any quick thoughts on that? We have about a couple minutes.
Yeah, my quick thought is that the studies all of us are referencing did actually use entire meals, not just individual foods. In the case of our study, we used 104 meals that were full meals, balanced meals and so on and so forth, and then we broke them down. I'm sure that the others had a similar approach.
Yeah, so we've been doing work looking at standardized meals so that we can look in a very controlled way, also looking at individual foods. but we've also collected data across the PREDICT studies now in 40,000 different meal configurations. And so we can tease apart, the dark matter that we often talk about. So not, the 26,000 different chemicals as well as the different combinations. So thinking about the food matrix, the nutrient-nutrient interactions. And I think that's so important, because there's no point just looking at a GI of 3 different foods. It's all about how it's combined. And I think this is how, from my understanding, is how many of these personalized nutrition approaches are considering it in terms of the composite meal and not just the individual foods.
All right, thanks.
And I just I just have to say that, like, I think that from my perspective, like, it's really hard for some patients to understand that we're talking on a meal and not on a food, but we also used a combination of meals and we scored all the meals. I couldn't show it in my presentation, but using the score, it's much easier to look on the whole meal and not on a specific food.
Okay, well, thank you for your insights. Once again, we have more questions than time, and thanks for your presentations. We're really, I think, one of the goals here is to kickstart a general discussion overall. So thank you again for your time and your insights.